Dear students, in our last lecture, we have discussed about the steps of target sites for antiviral drug action. So the 7 steps are here, where we can these are the 7 steps. All the animal viruses mostly follow. And the steps are adsorption, penetration, uncoating, transcription, translation, assembly, and release of the new virus. So these 7 steps can be targeted to inhibit the infection of any animal viruses that are causing different kind of infections in our body. To combat with different sorts of viruses, researchers have developed several synthetic molecules, and these molecules are mostly analogues of ribonucleosides and deoxyribonucleosides, nucleic acid base or their derivatives. This may include into the nucleic acid, usually the DNA, then interfere with nucleic acid function and selectively inhibit viral polymerases, not the function of the host cell polymerases, only selectively target and inhibit the function of viral polymerases. Now we are going to discuss about the classification of antiviral drugs. Antiviral drugs are classed in 4 different categories. The first class is anti-herpes virus, means the antiviral drugs developed against the herpes simplex virus. So in the right hand side of the table, you can see what are the drugs can be used for as anti-herpes virus: acyclovir, valacyclovir, famiciclovir, ganciclovir, etc. can be used. In case of anti-influenza virus drug, amantadine, rimantadine, oseltamivir, zanamivir can be used. The third class, or third category, they are anti-hepatitis viral drugs, or maybe non-selective antiviral drugs. These are subclassed into primarily for hepatitis B and primarily for hepatitis C. The drugs that are specific primarily for hepatitis B are lamivudine, adefovir, tenofovir, etc. And the drugs that are selected primarily for hepatitis C as hepatitis C antiviral drug are ribavirin and alpha interferon. The fourth one is specific for the retroviruses. So they are classed in 6 different categories. The first one is nucleoside reverse transcriptase inhibitor, and the drugs are zidovudine, lamivudine, abacavir, etc., tenofovir. The second one is non-nucleoside reverse transcriptase inhibitors, the reverse one, or the opposite one, and the drugs are delavirdine, nevirapine, etc. The third subclass is protease inhibitors. The drugs under this category are ritonavir, atazanavir, indinavir, lopinavir, etc. The fourth class is the fusion inhibitor, or the entry inhibitor. Here the drug is envovitide. The fifth one is CCR receptor inhibitor, and the drug is maravidoc. The sixth one, the last one, is integrase inhibitor, that is preventing the viral DNA to integrate with the host cell DNA, and the drug is raltegravir. Now, one by one, we will discuss about the few antiviral drugs. The first is idoxuridine or trifluorothymidine. Both the drugs are derivatives. You can see they are nucleoside analog. In the first picture, you can see a substitution of iodine. In the second picture, you can see 3-fluorine substitution. That is why this is trifluorothymidine. So these nucleoside analog, they are the modified form of deoxyuridine. Incorporation of such an unnatural, means these are the synthetic molecule, unnatural nucleoside 5' triphosphate that are called NTPs into the nascent nucleic acid synthesized by the virus particle prevents the further growth of the elongating RNA or DNA chain. Means they are actually inhibiting elongation of the viral RNA or viral DNA. So this particular process is known as chain termination. Means they are inhibiting elongation of the chain, elongation of RNA or viral DNA. So these drugs inhibit multiplication of herpes viruses mostly. They are used for topical treatment of herpes simplex keratitis disease, but due to having toxicity, they are not recommended for systemic use. Only topical use is permissible. So here you can see how these nucleoside analog, they are entering. So normally in the lipid bilayer of cell membrane, they have nucleoside transporter. But these nucleoside analog will enter within the cell cytoplasm via passive diffusion like the nucleoside. And these nucleosides then undergo sequential phosphorylation by using host kinases. So here you can see, in picture you can see one particular nucleoside is converted into mono, di and trinucleotide, triphosphate nucleoside. Means it is actually converting into a nucleotide. And when the nucleoside is converted into a nucleotide, a triphosphate molecule, then that particular molecule will be active. That is what is called a bioactive molecule. Here in the green color you can see the bioactive nucleoside triphosphate inside the cell of the intracellular compartment of the host cell. So this nucleotide 5' triphosphate or NTP analog of the antiviral, which antagonistically competes with the natural NTPs for binding or recognition of the viral RNA or DNA polymerase. So here there will be a competitive inhibition. This process is a competitive inhibition. So there is a competition between the natural NTPs and the antagonistically and the synthetic nucleoside 5' triphosphate analog of the antiviral drug. So these antiviral drug will bind with the RNA or DNA polymerase. Hence, no more elongation will happen. Like the reverse transcriptase, Same function is happening there in HIV also. So they are actually terminating chain elongation, and in this fashion they are inhibiting the proliferation of the virus or replication of the virus. The second one is vidarabine. This is also a synthetic purine nucleoside analog. This particular molecule inhibits herpes simplex virus and variola zoster virus multiplication. And they are also acting like the previous one, also acts as chain terminator or inhibit chain elongation, inhibits the viral DNA polymerase more than the host DNA polymerase. That is very important because they specifically bind with or recognize the viral DNA polymerase, not the host DNA polymerase. That is why only the replication of the virus, irrespective of the RNA or DNA, it will be inhibited, not the host cell DNA replication will be inhibited. What are the uses? Uses are herpes simplex keratitis, topical use, and herpes simplex encephalitis. This is intravenous mode of administration. Next one is acyclovir or fanciclovir. These two antiviral drugs are also synthetic nucleoside analog. They are actually guanine linked to an open ring analog of ribose and deoxyribose. They are either thymine or cytosine derivative. So mostly they are also functioning in case of herpes simplex virus and variola zoster virus. Thymidine kinases. So here these NTPs are also forming during function, and topical or intravenous in mucocutaneous herpes simplex in immunocompromised host and also in genital herpes simplex infection they are used. So what is their mode of action? So acyclovir first utilize ATP using herpes simplex virus thymidine kinase. So phosphorylation is going on. So first acyclovir monophosphate is formed utilizing the herpes simplex virus thymidine kinase or variola zoster virus thymidine kinase. And then human kinases will come into play and convert the monophosphate into triphosphate. And then this triphosphate will competitively binding with HSV or variola zoster virus VZV DNA polymerase and will incorporate into the viral DNA, finally inhibits the herpes virus DNA polymerase competitively. So here you can see that they are actually stopping lengthening of the DNA strands or elongation of the DNA strands. Tencyclovir, ganciclovir, these are derivatives, similar kind of drug like acyclovir. And they are also maintaining the similar kind of mechanism or similar kind of mode of action. Next one is amantadine and rimantadine. These two drugs are used mostly in case of influenza A. They are very much effective inhibitors of influenza A multiplication. How they are functioning? They actually bind to and block the H channel and prevent the M1 protein, that is a fusion protein, the M1 protein, from dissociating from the nucleocapsid. So affect penetration and uncoating. Also inhibits budding and virus particle release. So This is approved by FDA for prophylaxis of versus influenza A, but it has some side effect in central nervous system. So amantadine have more side effect rather than rimantadine, and this particularly inhibits the replication of each of the subtypes of influenza virus. So H1N1, H2N2, H3N3, so they are active against all these subtypes of influenza A virus. But they have minimal or no activity against influenza virus B, as this virus contains a different protein other than M2 in its membrane. So actually they are acting on targeting the M2. So as because this kind of protein, M2 protein, is not there in influenza B, so these kind of molecules are not active. So here you can see the scheme, how they are actually functioning. So mostly amantadine or rimantadine, they are actually functioning on uncoating, targeting the M2 protein and prevent uncoating. So due to this, the viruses cannot carry on further. So we are actually inhibiting, not inhibiting its endocytosis, but we are actually hindering the virus genomic material or genetic material to come out to the host cell cytoplasm from the endosome. So this is how they are actually functioning. Next one is oseltamivir. Oseltamivir is a neuraminidase acting against neuraminidase. So we know that neuraminidase has a function that is actually helping the progeny virus or progeny virion to come out from the infected cell and spread the virus to other normal cells throughout the body. So by inhibiting the function of neuraminidase enzyme, they are blocking the release of the virus, new virus, or progeny virus, from the infected cell. So there is no proliferation, there will be no infection further. So the uses are prophylaxis in the treatment of influenza virus A, swine flu, bird flu, along with influenza virus B, so that we can compare that oseltamivir is a more potential antiviral drug rather than amantadine or rimantadine.